All right, there's a little lull in the conversation. I'm going to take it. Okay, welcome to microbiology. Some of you guys are going to have to get here a little earlier to get a good seat. It's great to see so many people here. I was just thinking a year ago, teaching in this classroom, and everybody had their masks on. So it's quite different today. I'm Professor Leonard, so you're all in the right place. We are going to do the little syllabus thing and get to some content today, but I thought maybe I'd start out with telling you, like, I know why you're here. You're here because, you had to take this class, probably a lot of you. Hopefully some of you are interested to begin with, but you might wonder why I'm here, and I don't know, maybe you don't really care, but I thought I'd at least mention what drew me here and what drew me here to talking about biology with you today. And I apologize, like I only taught one class and my voice is like getting a little crazy on me already. So just let me know if you can't hear me. When I was, just briefly, back in the late 1989, Exxon Valdez disaster, like before you were all born. But at that time, I was late in my high school years and I was visiting colleges, and I sat down to talk with what was a future college professor of mine. And she was trying to talk me into microbiology. And this accident actually hadn't quite happened. It was almost ready to happen. But she talked about bioremediation with me, the idea that we could use microbes to clean up our messes. And that was such an attractive thing to me that it drew me to major in microbiology. And I don't really know very much about this today. Like I didn't foresee that, but certainly there's lots of things in microbiology that really interest me. Here's, just the idea that we could use microbes to manipulate them to make our lives better. It's just a really cool thing. Things that we don't even, they're all around us and we kind of forget that they're there most of the time. So cleaning up radioactive waste, any kind of pollution spill that you can think of. And another topic that really interests me is, you have at least as many microbes in your body as you have your own cells. Sometimes they say 10 times more microbes than your own cells. And if we could understand which microbes should be there and which ones shouldn't, we could be healthier. We could manipulate that to, make us weigh less. They think asthma and even autism have roots in our microbiome. And we know so little about that. And it's just exciting to think that, as we learn more and more that we can, manipulate our own bacteria and other microorganisms to make us healthier. So I really like microbiology and I'm just fascinated by it. And I've taught this class for about 10 years now, in lab longer than that. So I'm still learning. And I hope that you become interested too. Just a little bit about me. What brought me to West Michigan, I followed my husband. Some of you know him. A few of you have him right now, I believe. He's Dr. Leonard's over in chemistry, teaches biochem. You can call me Dr. Leonard. I'm not actually a doctor though. So call me professor, and then you won't make other PhDs mad when just call me Professor Leonard. I graduated from Cornell and got a graduate degree from a nearby university, SUNY Cortland. And we have 3 kids. They're all grown up. 2 of them live on the East Coast. And our middle child there in the middle, she has autism and she lives home with us and works. And so that's going good. But life is a little bit quieter now that my other 2 are out of the house for me. So that's just, a snippet about me. I try not to tell too many stories. Sometimes I make fun of things that my son has said to me from time to time. But we really got to keep buzzing along in this class. We have a lot of content to cover. And I hope that you haven't heard too many horror stories about this class. I hopefully will convince you today that it's doable. So I'm going to give you some tips as we start out today. I'm pretty reachable. I'm over in that hallway in Henry where all the micro people are. If you're in lab, it's right down the hall from lab. I know some of you aren't in lab right now, and I do talk about lab a lot, but don't be nervous if you're not in lab yet. That's fine. Office hours, those are the times that you can be sure to find me in my office, but I'm there lots of other times. So if those times don't work for you, don't be afraid to set up another time. Come March, I start a little side gig of mine. I'm a assistant track coach for my high school. So after 3 o'clock, I'm a little harder to reach. But until March, that time works for me too. Okay, and we can do it by Zoom. We can do it in person, however you want. Some of you are already signed up for Top Hat, which is the way that we have you access the textbook. So good job. Some of you already gotten into that. We switched last semester to using an OpenStax textbook, which is a free textbook. And it's been kind of not a hard change, but a change that's required a lot of work on my part to switch over from our old textbook system. I like the new textbook pretty good. It doesn't have a lot of the sort of student helps that the old textbook did. So we're saving you a lot of money. The old textbook was about $100. The OpenStax textbook, you can access that for free. Why do you have to pay $30? Well, we've employed Top Hat to kind of help us. It's the platform by which we have changed that OpenStax textbook a little bit to meet our needs. And that's the way that we deliver the homework to you. So you have to buy Top Hat. It's $30 for a semester. If you have it for another class, you don't need to buy it again. If you pay, I think, $40, it's good for a year. But that's the way that there are questions at the end of every section in the textbook. The students that I asked students last year on their evals to say, oh, did you like doing the homework? Did you not like doing the homework? And it was kind of a mixed bag. Some of them said it was dumb. Some students said, yeah, this really helped me. So it's kind of a process. I spent a lot of time over break trying to make those homework questions better, trying to make them ask questions that I might ask you on a test, or certainly less questions that just kind of ask you about stuff in the textbook that I'm not going to ask you about. So trying to make that better and better. That homework portion of your grade, all students like that cushions your grade a little bit. So the way I have that set up is basically if you get 80% of your homework questions correct, then I give you the full 60 points of homework. So the idea is I don't want you stressing over every little question. Every once in a while, if a question's just right way out there, guess, okay? It's actually graded very leniently. Every question gives you a half. If it's a 1-point question, it gives you a half point just for trying. So it is quite lenient. Don't spend a ton of time on it, but it is one other mechanism to get you to review and to practice kind of basic level questions. They're not terribly clever questions. They're more like, what was this? Or what was that? You know, just kind of basic review type questions. But I hope will help you. You need to see things lots of different times before you really get it into your memory. The other thing you might notice is the first exam is worth a little bit less. I started doing that last year because I noticed there were some students that would really bomb the first exam, and then it's hard to come back from that. So I wanted to make the first exam worth a little bit less, and it covers a little bit less material. So we're going to have that right away. I think we have 4 classes, and then we're going to have an exam. So the idea is, let's kind of start you out hopefully on a good foot. If it doesn't go well, it is worth a little bit less, and think about how we can come back from that. The final is worth a little bit more. Okay, if you're doing better and better and better as you go through the class, then you want to be rewarded by that, right? So we'll talk more about that later. Okay, so that's the grading. So how can you do well in this class? I would encourage you to read the whole syllabus, because I go into a little bit more detail about some of these things on the syllabus. But hopefully you notice I posted some PowerPoints. Hopefully those might not get us through next lecture. I might need to post some more. We'll see where we're at. But those PowerPoints are just an outline. So you need to have some way to take some notes in class. I work off a tablet. That's probably maybe the best way, if you have a tablet and you can just write on notes. You might need to convert that PowerPoint into a PDF so it doesn't shuffle things around a little bit, depending on how you're doing that. A lot of people last semester would take notes off their laptop, print it out, you know, maybe 2 to a page. That's another legitimate way to do that. So let me know what works for you, or if something's not working, let me know. Maybe I can adjust a little bit. I have for every chapter posted learning objectives. So these are on Blackboard as well. It's kind of a long document. These are for the whole semester. I've tried to make my tweaks in between semesters here, and I hopefully won't have to shift these around too much. But I would suggest that you print these out. And like I said, there's a lot here. Inch by inch, life is inch, yard by yard, life is hard, right? So I would encourage you to kind of keep up with this as we go. You know, maybe have a certain couple times every week that you're going to sit and look through these and, you know, kind of think about it. Some students like to, it's a lot to write out the answer to all of them, but at least use this as a guide. At least look at this. I have terms that I think will be new to you in bold type. Okay, so that might be helpful. What I do when I make up an exam, I look at the learning objectives. I say, oh, yeah, this question covers this one. This question covers this one. So I kind of make sure that I covered not a bunch of questions about one thing and nothing about another thing. So that's one tool that we have. I would encourage you also to do that homework maybe within a few days of when we've covered it in class. I noticed some of you have started it already. And that's fine. It might take you a little longer to work through it because there may be some questions that I only cover in lecture and actually aren't in the textbook, probably not too many. But you'll be able to work through it a little bit quicker if I've already talked about it in class and then you do it later. Some students wait till 3 a.m. the night before the exam to do the homework. I would not encourage you to do that. You're not going to do as good a job on it, and it's not going to do you as much good if you're rushing through it. Okay, so kind of work through it. I would encourage you. Terminology. Part of your final exam is just going to be matching of terms. So it's really important that if I use a term or if you know, there's a term in your notes that you don't understand, don't just skip over it. Stop and take the time to understand what that means, because if you just kind of skip it over stuff, then it's going to build up. So learn the terminology. Study as actively as possible. So I talk about this in the syllabus. I read a book a couple years ago, how to learn like a pro, science-based tools to become better at anything. And basically this is written for college students to help them understand how to study better. So I gave you some pointers there. Basically when I was in college and I studied, I remember what I did. I read through my notes. And that's probably the worst way that you can study. Okay, that is not active. Your brain can just shut off. So some pointers here to how to study more actively. Pretend you need to teach it to somebody else, or if there's nobody else around, a stuffed animal or something. Like, teach your stuffed animal what's special about B cells, or what an opsonin is. Okay, you need to be as, what I mean by active is always checking to see if you know something. Another way you could do that, if you're doing some reading, okay, I just read that paragraph, stop and think, what did I just read? If you have no idea, then you need to go back and look at it again and then test yourself again. So as much as you can test yourself, the better. I do give you some practice multiple choice questions before the exam. It's not a lot. I really like to try to expand those number of practice questions that I'm giving you. So that's something that I'm working on. But it does help to study in groups sometimes, right? Sometimes that gets to be just a, kind of a social event, but it might help to find a few people that you can study with. Okay, so if you're sitting near some people that you don't know, I want to give you a minute and introduce yourself to who you're sitting around. So get to know somebody, all right? Get somebody's name. So I'll give you a minute. Talk to somebody around you that you don't know. So do you guys talk to each other? Oh, okay, good. Okay, good. I knew that wouldn't be a problem. Okay, don't be afraid to do that. Okay, lastly, I do record the lectures. So we are recording right now. I started doing that during COVID, obviously, because people had to miss for long periods of time. And I think it's really helpful that when you do have to miss, like if you do, don't come sick because, you feel like you can't miss anything. The recorded lecture is pretty good. It's not as good as being here though. So I would encourage you to come, just because then you can ask questions and you can, again, make connections with other people. It is better to be here, but if you can't be here, they are recorded. And I usually get them up the same day. Yeah, sometime the same day. So unless I have some kind of technological meltdown, those will be there. But again, it's better to come. I hope that you'll come as much as possible. Okay, stop me with anytime if you have questions. I just wanted to, quick rundown. What are we going to be talking about this semester? We start out about just basics about microbes. And I'm going to talk about what I mean by microbes in a minute. But structure of cells. There's a lot of framework for the rest of the semester. How they get their energy, how they grow. What does that look like when we're talking about microbes growing? How we describe populations, and genetics as well. Like how do they pass on their DNA? So a lot of basics in the first and second exams, And then we get maybe a little bit more practical and talk more about how we want to kill microbes. This course has a very clinical bent. So we are thinking mostly about, yes, microbes are bad and we want to kill them. It's actually not the case. Like I said, I'm really interested in ways that microbes help us and they're necessary for our health. But we do spend a lot of time thinking about how to kill them. So, use of disinfectants, use of antibiotics, how our body works, how immunology works to try to cut down on the number of infections. And then we end the semester called our bug parade, thinking about some very specific infections and kind of wrapping things in from the whole semester that, what makes those organisms successful. Okay, so that's a glimpse of what's ahead. Any questions, anything about the syllabus? Exams will only be in person. Like we're past respondents, like we're not doing that anymore. So I do give makeup exams. Some of the other professors and teachers don't do that. Just, if you got a good reason, make sure you let me know, and we got to try to figure out how to get you a makeup, hopefully within a couple days. I don't like to let things drag on too much. If you're sick for a long time or whatever, sometimes we will skip that exam and we'll double your final. Okay, I don't like to do that very often because that puts a lot of pressure on you for the final, but sometimes that's our best option. Okay. All right, so chapter 1, we're talking a little bit of history here to begin with. I always like to try to tell you what I'm going to tell you before I tell you. Other thing I didn't mention, Top Hat, most times people use Top Hat for clicker questions, and I have used clicker questions in class before. I love doing that because it just makes class a little bit more interesting. I have not really found a way to do clicker questions and record lectures at the same time. So, and plus it kind of slows me down when I'm writing in content. And so we won't be using it for clicker questions. At some point I might change my mode on that, but in case you were wondering about that, some of the other professors do. Okay, so we're going to start with some history, taxonomy. Hopefully we'll talk a little bit about why that's important. It's a little dry, but we won't spend too much time on that. And really then just kind of get into what do I mean by microbes and what are the things that we're going to study this semester. All right, so first glimpse of microbes. The guy that's given the most credit for producing the first simple microscope, his name is Antony von Leeuwenhoek. I don't think I gave myself enough room here. That's a little bit fat. There we go. Okay. So a Dutch cloth merchant, Antony van Leeuwenhoek. Again, he's given, there's some discussion over whether his was really the first one or whatever, but he's really much given credit for the first simple microscope, and that just means a microscope that had only one lens in it. Okay. And like I said, he was a cloth merchant. And that seems like kind of a strange profession for somebody that develops a microscope. But he was Dutch, 1700s time frame there. Not too worried about dates, but just to kind of get a time frame. Dutch people today have kind of the stereotype of being kind of thrifty, right? So he wanted to make sure that if he was paying a lot of money for a fine bowl of cloth, that he was not being cheated. So that's what led him to produce these lenses that would enable him to look really closely at the cloth and make sure it was the good quality that it is. Okay, I'll come back to this slide in a minute, but here's a picture of von Leeuwenhoek. And here is a picture of that first microscope. Looks bizarre, right? The lens is right here in the center there, and then there's kind of a little sticker here where he could impale like a fly or whatever he was looking at. And he could kind of hold it up above him like this and look through that one single lens. Looks primitive, but they think that it enabled him to have about 100 times magnification, which is equal to about medium power on our microscopes in the lab. So he was getting some good magnification. He learned how to make these really high quality lenses. And he looked at all sorts of things. He looked at water and blood and semen and just all kinds of crazy things. And he wrote up what he saw to publish. And so he was a very kind of recognized scientist of the day. He called the things that he saw wee animalcles. Okay, so little beasties, sometimes he called them. So all sorts of things. One of his contemporaries, who was born in England, was Robert Hooke. And he is given credit with making the first compound microscope. And that just meant that there was 2 or more lenses in his microscope. Okay, and Robert Hooke is also credited with the first one to use the term cell. That's kind of the basic unit for all living things. This guy here that I didn't mention, that's Galileo Galilei. He did some early work on microscopes, but he's better known for his work with telescopes, so we're going to skip over him. Robert Hooke, here's a picture of his, or a drawing of his first microscope. So apparently there was a couple lenses in there. You can see a candle here. That had to have been the most difficult thing to get light focused in just the right spot. Very difficult before we had a light bulb to do that. But he looked at some cork and he saw that they were it was compartmentalized into little rooms, and again, he called those cells. And then we'll skip forward about 100 years to what's known as that golden age of microbiology, about 1850 to 1910, where just lots and lots of advances were made in the field. And really lots of advances were being made scientifically in many other fields in that time period as well. But this is kind of the beginning of the work of Louis Pasteur. He was a French chemist. We'll mention him from time to time. We're going to kind of walk through one of the experiments that he did where he first kind of came up with the pasteurization, a way to heat up things, liquids to cut down on microbe life. We're going to talk about that in a minute. He's also known for making a couple of vaccines, the anthrax and the rabies vaccines. Okay, so that's Louis Pasteur. He was French. The other player here, Robert Koch, he was German. They're roughly contemporaries. They actually helped with the war effort during the Crimean War, and they were on opposite sides of that war. So I don't know if it was for that reason, but they famously hated each other, these 2 scientists, even though they worked on lots of the same problems. Robert Koch is kind of best known for understanding the causation of disease. So he worked out what microbe caused anthrax, cholera, and tuberculosis. The TB one is really an important one because that was a huge scourge at the time. And at that time, people thought that tuberculosis was a cancer because it was really slow acting and it tended to run in families. And so that was really a big deal that it was actually an infectious agent. Okay, so we'll talk more about Robert Koch. He's famous for developing these postulates that had to be satisfied to understand what agent actually caused the disease. So we're going to talk about that a little bit later. But from time to time, I like to bring up famous experiments that took place just to kind of, I know you all know what the scientific method is, but just to kind of illustrate that and remind us how important it is to have good controls and to only kind of change one variable at a time. That's kind of the scientific way. So this talks about how Pasteur applied the scientific method to understand fermentation. So, like I said, Pasteur was actually trained as a chemist, even though he kind of went more into the medical fields later. And around 1850, the French wine industry was struggling because their wine kept getting spoiled, and they didn't really understand what it was that made grape juice turn into wine. So they didn't know how to fix the problem. So they consulted with Louis Pasteur, and he kind of set up a series of experiments. So first thing that he understood was when grape juice was fermenting and you looked at that under the microscope, you could see both yeast and bacteria. How could you tell the difference? Yeast are a lot bigger than bacteria, so it's pretty easy to tell the difference there. So he set up some experiments. And had some hypotheses. So first hypothesis that grape juice just spontaneously ferments and turns into alcohol, wine. So to test that, he took some grape juice, he boiled it, and so everything was, weren't any microbes in there, sealed it up, and then opened it up at a later time, and it was still grape juice. Nothing had changed. So reject hypothesis one. It's not spontaneous. Is it something in the air that ferments grape juice? Well, Pasteur had developed these famous swan neck flasks that he used to disprove spontaneous generation. We're gonna skip over that and not talk about that. But with this swan neck flask, he could have the grape juice open to the air, but anything that was in the air would settle out and kind of settle in the bend there of that swan neck flask. So he could leave it open to air, he let it sit, and the next day, no fermentation. Still no microbes in there, no alcohol in there. Okay, so it wasn't the air. So then he hypothesized maybe it's the bacteria that's doing the fermenting. So he inoculated that grape juice with bacteria, and then the next day tested what was in there, and he found that there's lots of more bacteria, and it was more of an acidic quality to the grape juice. And so in fact, you'll see this as you move through the lab, but when bacteria are using sugars, they almost always produce acidic end products. So the grape juice, he had to kind of modify that a little bit. The bacteria are going to turn the grape juice into acids. So then he said, well, maybe it's the yeast that's doing the alcoholic fermentation. He inoculated the juice with yeast and then found alcohol present. So it's the yeast that are doing alcoholic fermentation, what you want. So he came up with this process to heat up the grape juice sufficiently to kill everything, and then what's called pasteurization, and then just added just the microbe that he wanted, the yeasts, to do the alcoholic fermentation. It was the bacteria getting in there that was spoiling the wine. And we don't use pasteurization for wine anymore because it does alter the flavor a little bit. I think they do that chemically mostly. But of course, we use pasteurization to control the microbe contents and lots of other things, mostly milk. So we'll talk about that more later on in the semester when we're talking about controlling microbial growth. Some other famous people, and again, hopefully I have this spelled out pretty closely in those learning objectives, the people that I want to make sure that you just kind of understand basically what they contributed. John Snow is called the father of epidemiology. So that's kind of a hot topic in the pandemic years. Epidemiology is the study of how not just infections, but how diseases spread. And it's a lot of question asking, and a lot of statistical analysis. But what John Snow did, there was a big cholera outbreak in 1854 in London. And he had his suspicions that the cholera was caused by contaminated drinking water, but he didn't have any way to prove that. What he did, he took a map of London and then he went around to every household and asked questions. He said, do you have anybody sick with cholera? Where do you get your water? Where do you get your food? Where do you, whatever, where do you do your laundry? Whatever, and everything. And you can't see it very well on this map, but he kind of made a note of all those, made a note on his map of where there were sick people. And he noticed after all this information gathering that the cholera outbreak was centered around the Broad Street water pump. That's where people went to get their water. So he took the handle off the pump so people couldn't get water in there anymore, and the outbreak subsided. So it's kind of a simple simple answer to a very complicated question, but they didn't understand really at that time that that there was just probably some sewage contamination right near that pump, and that's what was causing it. Okay, so that's John Snow thinking about preventing infectious disease. A number of people we can think about their contributions. We have the first American here on the list. A lot of the early microbiology and advances were made in Europe. But Oliver Wendell Holmes Jr., his son went on to become a Supreme Court justice. So he may be the one that you've heard of more. But Oliver Wendell Holmes Sr., he was a physician and a poet, and he did all these other things. But he wrote up a paper about puerperal fever, otherwise known as childbed fever. And he just made, he looked at a lot of different data. And he noticed that a lot of women were dying in hospital. They gave birth in hospitals rather than if they gave birth at home. And he advocated for hand washing as a simple way to just improve the hygiene. So he was kind of really, I don't think talk about him in your book, but he was kind of one of the first ones to advance this notion that, hey, hygiene might be important. It was kind of before we were just kind of starting to chip away and understand germ theory, the theory that it's microbes that are making us sick, for a lot of infectious disease, or for infectious disease. Probably more famously, Ignaz Semmelweis. He was a Hungarian physician, and he, again, noticed that women in hospitals were getting this childbed fever after they gave birth. And this was women were dying of this. He noticed that physicians would go right from doing autopsies up to the delivery ward, straight away, and weren't washing up. So he advocated, again, hand washing. Unfortunately, his colleagues didn't appreciate him suggesting that they were the cause of women dying. And so he eventually lost his job and died in an insane asylum. So it takes a while for things to catch on. Couldn't really explain what it was. Didn't really understand that it was bacteria and viruses doing this. Florence Nightingale, she is famously a nurse in the Crimean War, and really kept very careful notes, kept very strict hygiene of her patients coming off the battlefield, and good statistical analysis showing that good hygiene saved lives. So again, this is kind of starting to be more and more accepted that hygiene is important. And a little bit later here, Joseph Lister, he was an English physician that went a step further from hand washing. He said, hey, physicians need to wash their hands in carbolic acid, which is phenol. And they sprayed phenol in operating rooms to try to cut down on infections during operations, and drastically that worked. Now, unfortunately, phenol has some side effects, and there's reasons that we don't do that anymore. But again, kind of big steps in improving hygiene, and that goes a long ways into preventing infectious disease. Another really important tool we have in preventing infectious disease vaccines. We're not going to talk about treating here just yet. We're going to save that. But Edward Jenner is famous for developing the first vaccine. It happened to be to smallpox. So this is early. We're going back here, 1796. He's given the credit for this, but again, there's some suggestion that maybe this wasn't his idea and other people thought of it and didn't, get the credit, didn't write up the paper or whatever. But he noticed that milkmaids very rarely got smallpox. And because they often contracted a disease from cows, a related disease called cowpox. So he did an experiment. He took a child, not sure who this child was, but he intentionally inoculated him with cowpox, let that heal up. You just take the crusts from a lesion of cowpox and he inoculated them. And then a few weeks later, he took the scabs from somebody sick with smallpox and inoculated the same young boy. Not exactly the way we do clinical trials today, but lucky for him, the boy survived. And this is a greatly improved way to keep people from getting smallpox than the variolation, what they did before, taking somebody with smallpox scabs and inoculating people. That still carried about a 1% fatality rate. So inoculating somebody with a milder disease was a big improvement. And we've been improving on vaccines ever since. All right, so a little bit more history to begin our section here on taxonomy. What is taxonomy? That sounds kind of boring. Taxonomy is really a science that's always changing as we get better information. But we could think about taxonomy as classification, identification, and nomenclature all together. So classification, that just means assigning organisms into groups. Okay, so kind of trying to understand the relatedness between different organisms and putting related organisms all in the same group. Identification is understanding which group an organism belongs to. And then nomenclature, that just refers to naming rules. We have to have some rules about that because we can't have everybody thinking up their own name for an organism. Okay, that would make a lot of confusion. Okay, so the father of taxonomy, the one who really kind of got a bunch of rules started, that's Carolus Linnaeus. Okay, so we're going back. His life overlapped a little bit with von Leeuwenhoek. So we're going back to early 1700s again here. He was a Swedish botanist. And again, he thought up a lot of these rules for taxonomy that we still adhere to today. And he's also the one that thought up of the different levels of taxa. So you probably heard these before: kingdom, phylum, class, order, family, genus, and species. Now you don't really have to memorize those. I remember them, the mnemonic. King Philip comes over for grape soda. Probably other mnemonics. But that, again, don't really need to know the order of that. We do talk a lot about the genus and species, okay, because that's how we name organisms. All organisms have a unique name that includes both the genus and the species. And Linnaeus said all organisms need to be categorized in these different levels of taxa. Now, he said 2 kingdoms, plants and animals. Right? We have many more kingdoms than that today. Okay, so we've been revising this ever since the 1700s, but he put all life into different groups based on shared characteristics. So, you know, with organisms, you know, like mammals and plants and fungi, that's a little bit easier to do with things that you can see. You know, you might use whether they have fur, whether they have feathers or whatever, shared characteristics. He classified species as organisms that interbreed. Now, that's tough with microbes, right? Bacteria do not reproduce sexually. It just one bacteria cell splits and becomes two. There's no interbreeding. So today, we define a bacterial species, a species that have about 70% DNA homology. Okay, so we have to kind of look at the sequence and decide that way. Or that's one of the ways that we decide. Okay, let's see, what else did Linnaeus do? Here's a little picture of Linnaeus as a younger man. He's also noted for making up the rules for naming organisms. And he said, we're going to use the binomial nomenclature. Oops. And that means, okay, that means that every organisms need to have a two-part name. Okay, so for example, Homo sapiens, that's humans, that's the genus and the species. Genus Homo sapiens species. There's a flower that's named in his honor, Linnea borealis. Okay, so he said these scientific names, they should all be, you know, he was Swedish. What if he said everything needs to have a Swedish name? Okay, people in the world maybe went to other places in the world wouldn't have bought into that. So he said the genus and the species should be Latin. Okay, and what that means, we kind of throw an Latin suffix on it most of the time. And he said that the genus, like Linnea is a genus, Borealis is a species, the genus should be a noun and the species should be an adjective. Now those rules aren't strictly followed, but in this case, Linnea, it was named for a person, so that's a noun. Borealis, I'm not actually really sure what that describes. Something like Escherichia coli. Escherichia was named for Theodore Escherich, the man who discovered the organism. And coli just means of the colon, so it is descriptive. But that's, again, that the noun and adjective isn't strictly followed. Notice here that he has italicized the genus and the species. Okay, so we have grammar rules, and I'm kind of stickler about this. Your lab professor may be a stickler as well. If you're typing out a genus and species, you should write it in italics, and the genus should be uppercase and the species should be lowercase. Okay, if you're handwriting a genus and species, it's correct to underline because you can't write in italics, right? So it's correct to underline. And again, let me write that out. Genus, uppercase, and the species, lowercase. And so you're microbiology students, you're going to be writing bacterial names a lot. Just get in the habit of doing that grammatically correct. It's kind of a pet peeve of mine. You see it in the newspapers and stuff like that where they don't follow those rules, right? And that always bugs me a little bit. Oh, and sometimes people ask me, well, when can I just write E. coli? When can I abbreviate that genus? Well, if you're, say, writing a scientific paper, the first time you mention an organism, you should write the genus and the species out. but then every time after that, you can abbreviate the genus. So something like E. coli, that's so common that you just might see it referred to as E. coli without the full name. But technically, you're supposed to write it out at least the first time. Okay, so we have been, tweaking taxonomy ever since the 1700s. This is Haeckel's contribution. I'll write this a little bit bigger here. He added a couple of more kingdoms. Remember, Linnaeus said plants and animals. He added the protist kingdom as well as Monera or Monares, which included bacteria, basically. So that was, about 100 years later, mid 1800s. And then Robert Whittaker, he added an additional kingdom, the fungi. Okay, so up to that time, all the fungi were in the plants. So what's the difference? There's a lot of difference, but what's the main difference between plants and fungi? Yes, good. So plants photosynthesize and fungi don't. Good. So that was kind of what I remember in my high school biology, learning Whittaker's taxonomy. We had to memorize those kingdoms. Well, that's not actually the most correct way to present taxonomy today, because in the 1970s, Carl Woese and his student George Fox, they were interested in the science of taxonomy, and they were sequencing many, many different organisms. And in the 1970s, to sequence any kind of nucleic acid was a really laborious process. You had to run gels to get, one base pair at a time. So it was a lot of work. And they actually were sequencing ribosomal RNA instead of DNA. DNA is double-stranded. It's a lot more complicated. Ribosomal RNA is single-stranded. So that made it a little bit simpler. I was reading it would have taken them another 10 years if they would have decided to sequence DNA instead of ribosomal RNA. But ribosomal RNA is pretty important. Where do we find ribosomal RNA? Not a trick question. In the ribosomes, okay? And what do the ribosomes do? They make proteins, yes. So it's a very important structure that all cells have, okay? And ribosomal RNA is highly conserved. What I mean by that is if something goes wrong and the ribosomal RNA is not transcribed properly, then probably that organism will die. Because it's really important that organisms can make proteins and make them correctly. So it's highly, so sequence of ribosomal RNA is passed down with pretty high fidelity from generation to generation. Okay, and so he sequenced the ribosomal RNA to determine phylogenetic relationships. That's another way to say evolutionary. He wanted to see which organisms had a closest common ancestor, just to try to understand, what groups they should be put in. And what he expected when he did this with a lot of different organisms, he thought he would have two kind of big groups, the prokaryotes and the eukaryotes. Okay, so remember, prokaryotic organisms, how they're different from eukaryotic organisms is that they don't have a membrane-bound nucleus. Okay, prokaryotes do not have a membrane-bound nucleus, and they don't have membrane-bound organelles. Okay, they do have ribosomes. Ribosomes are not enclosed in a membrane. As we'll see when we talk more about this later, the ribosome is actually a small subunit, a large subunit, and has to come together when it's making proteins with that messenger RNA sequence involved as well. So those are not membrane bound. So he thought that there would be two big groups of organisms. That's not what he found when he did all these sequences. He actually found three different categories, or three sequences that shared a huge amount of homology. Okay, so he proposed that above that kingdom taxonomy, there should be a level called domain. Okay, and he suggested calling those domains the eukarya, the bacteria, and the archaea. Okay, so if we went back here to Linnaeus's list here, now we include another level above kingdom called domain. Okay, so there's only 3 domains. Okay, and so this is this tree of life, this phylogenetic tree of life that he came up with, based on, again, the ribosomal RNA sequencing. So we're over here, we're in the animals and in the eukarya. And this is not something to memorize. I don't have a lot of these are all correctly called kingdoms now. I don't have those memorized either. But where was I going with that? So we're over here with the animals, the fungi, the plants. All of these are different, what we would mostly refer to as protozoa. Some of those lines are a little blurry between protozoa and fungi, the slime molds. We'll talk a little bit more about the eukaryotes at a later lecture. Then the bacteria and the archaea are what are also known as prokaryotic cells. The archaea, we don't mention these a lot in this class. A lot of they're all single-celled organisms. A lot of them some of them at least are known as extremophiles. So they live in crazy environments like the Great Salt Lake. That's a halophile, an organism that likes high concentrations of salt, or in places where it's really hot or it's really cold. But there are some archaea that probably live as normal flora, normal microbiota in your body as well. But what we do know is that we haven't found any organisms that are considered archaea that cause disease. So that's why we don't pay as much attention to them in this class. Mostly what we're focusing on are the bacteria, and we'll talk a lot about gram-positives. I think a lot of the gram-negatives that we talk about are in the proteobacteria, but there's a lot of different kingdoms there. One other thing that I'll point out about this tree of life, if you look at the closest common ancestors, you'd really kind of expect the 2 prokaryotes, the bacteria and the archaea, to have the closest common ancestor. But look, here's the archaea. It has its closest common ancestor with some early organisms in the eukarya. So that's kind of surprising. And if you look at structures from archaea, some of it is like a lot of its metabolism is very similar to the bacteria, but then some of the way it does its genetics is a little bit more like the eukarya. So, and then it has some things that's kind of special all its own. So, okay. So we're going to refer to that from time to time. Thinking about what else we use to categorize organisms, kind of the first thing that you think of is something that is really that you can just see, an observable characteristic, and that's the shape of the cell. Okay, so some terminology that goes with that you'll want to get familiar with. I feel like that's just not very big up there. A round cell is called a coccus. Okay, or plural, many round cells, we would say are cocci. Okay, so round cells, rod-shaped bacterial cells are called bacillus, or again, plural, bacilli, many of them. If you're in lab, that's probably all you're going to see, cocci and bacilli. Those are the kind of most common morphologies of cells. But there's a lot of other crazy things out there. There's vibrios. Those are kind of comma-shaped cells. A coccobacillus is kind of halfway between a round cell and a rod-shaped cell. Really stubby rod-shaped cell. And then there's some helical morphologies, the spirome and the spirochete. We'll tease out the difference between those a little bit later, but they move in a corkscrew type way. We have different flagella that make them move that way. In addition, there are some cells that are considered pleomorphic. And that just means they're not any shape. They can be lots of different things. Sometimes that's because those cells don't have a cell wall. And the cell wall is kind of what imparts the shape. Okay, so there is this one genus of bacteria that don't have cell walls, and they're considered a pleomorphic morphology. Okay, so that's kind of an easy thing that we've always used as part of taxonomy. There are lots of other things that we could use and that are used. Not anything in specifically to memorize here, but in addition to the cell shape, how cells arrange themselves, and we're going to look at that a little bit more closely later, but meaning, do they tend to form clusters? Do they tend to form chains? Gram stain, we'll talk more about that later, but it has to do with particular cell wall structures. Bacteria are usually either one particular structure or the other, the gram positive or gram negative. And then lots of different biochemical things. Like for bacteria, we're putting into groups, we're interested in what kind of enzymes they can make, or what kind of sugars they can use for metabolism, or maybe what kind of metabolism do they do? Do they produce a gas, or do they not produce a gas? So these are just some examples of some tests that you're going to run in the lab to kind of try to ascertain and kind of characterize different organisms. We could do lots of molecular techniques too, things like polymerase chain reaction, PCR. ELISAs are where you take antibodies to try to see if they stick to certain structures. DNA sequencing, so there's a lot of molecular techniques that we could use too. That usually are not as good as the more like biochemical things, but sometimes we have to rely on those things as well. Okay, and there is one kind of, where do you go to find out all the information that we have about a particular organism? That's called Burgess Manual. So that's kind of the go-to if you want to know everything there is to know about a species of organism. And we have a couple old volumes up in the lab, and I think you can access that online, but that's just sort of like the go-to. Okay, so also I want to talk about today what kinds of microorganisms we're going to be studying. Okay, so obviously in microbiology, we're studying things that are too small to be seen with the naked eye. So we talk a lot about bacteria, but we're going to mention some other different kinds of organisms as well. Like I said, the bacteria and the archaea, those are the two domains of prokaryotes. And then in the eukaryotic organisms, there's microscopic protists, that includes algae, as well as things like paramecium and amoeba that move around. There are microscopic fungi. Of course, there's fungi that's not microscopic that we're not as interested in. Helminth worms, things like tapeworms. Of course, tapeworms are not microscopic, but sometimes there are helminth worms where part of their life cycle is microscopic. So we don't get into helminth worms in this class, but we do have a parasitology class that's taught every fall here at Grand Valley, which is very good if you're interested more in that. And then, of course, there's the viruses. I haven't even talked about viruses yet. They are acellular. So that means they do not have a structure of cells. So they don't have all the things that other cells would have. They might have some of those things, but not all those things. Okay, so I'll give you some examples in a minute. When we are thinking about learning about microscopic organisms, we want to kind of understand what units we're going to use to describe how big they are. Because if you're trying to measure something that's really small, you want to have anything you're trying to measure, you want to have a unit that's smaller than the thing that you're trying to measure. Okay, if you're trying to measure your foot in kilometers, it's going to be like a 0.0000, really cumbersome number. So we want to use small units. You might not be familiar with using some of these small units before. You all know what a millimeter is. Like that's 0.001 meter. Okay, so in metric, you move that place, that decimal place. A millimeter is a 1/1000th of a meter. Okay, so you move the decimal 3 places over. And well, how that's pretty, you can see things a little bit smaller than a millimeter, but if you picture a ruler, you know, a millimeter is like the very smallest designation on a ruler. And then if you look at like a pollen grain or a human egg, it's going to be a little smaller than that. This unit, a micrometer. Okay, whoops, 1 micrometer. Sometimes we call that unit a micron. Okay, this unit right here, this Greek U, I think, means micro. Okay, so that means a millionth of a meter. So this is 1, 2, 3, 4, 5, 6 of a meter, or 1 × 10⁻⁶ meter. Okay, the micron is a very useful unit because lots of bacteria cells are about a micron in diameter. Okay, so bacteria, most of them in general, about a micrometer. And that's about the same size as some of the bigger organelles in eukaryotic cells, like a mitochondria. A unit less than that we'll refer to every once in a while is a nanometer. So that's not a millionth of a meter, but a billionth of a meter. So 1 × 10⁻⁹ meter. Okay, so 3 more decimal places. So that's more helpful when we're describing the size of viruses. Okay, and this is kind of a fun website to visit. How big is it? So it starts with something big, and then it zeroes in and zeroes in and zeroes in. It kind of helps you appreciate really how small microbes are. So peek at that sometime. It's just kind of for fun. All right, so just again, giving you some examples of the kind of things that we're going to study this semester. Like I mentioned before, which I hope is reviewed, prokaryotes are going to lack a membrane-bound nucleus, as well as a lot of organelles, membrane-bound organelles. That's the bacteria and the archaea domains. You can't tell by looking at one of these cells whether it's a bacteria or archaea. You likely can't tell. Some of the archaea have some really bizarre shapes to them that you might guess. We'll get into a little bit later some of the structural differences, but just some examples. This Deinococcus, that's kind of a weird looking bacteria. It's actually a bacteria, and it's actually one that breaks down radioactive waste. Here's Staph aureus. You're going to be using that a lot if you're in lab this semester. We'll be talking about in here quite a bit too. And here's an archaea and a methanogen. It's an organism that actually produces methane as part of its metabolism. So kind of a strange one. And then the eukaryotes, we do spend a little time talking about fungi, a few fungal infections. This one that I have pictured here is kind of a famous one. That's the mold that actually produces penicillin. It's Penicillium chrysogenum. Okay, so we're thankful for that. Here's some yeast, some Saccharomyces cerevisiae. If you like alcoholic beverages, you can be thankful for that. And then protists, again, we spend really a very small amount of time on the protists. Things like amoeba and paramecium. There are some that are parasitic, so we'll delve into that a little bit. The algae, I mean, they're the basis of a lot of marine food webs and produce a lot of oxygen for us, but They are for the most part not pathogenic, so we don't really talk about those too much in this class, but there's freshwater algae, marine algae, sometimes called diatoms. Okay. And then again, the helminth worms, you know, fascinating, but we just don't have time to really get to those in this class. Some stages are microscopic. Viruses, we will talk much more detail about viruses, but like I said, they're acellular, don't have a cell structure. And in fact, here's a nice diagram of that diagram I've used for years and years. It's the SARS coronavirus, actually. They are basically just made of some nucleic acid, either DNA or RNA. There's no viruses that actually have both of those things. They always will have this protein coat around that nucleic acid, and then sometimes they have a membrane around them. Okay, so they're fairly simple, and they infect all types of cells. So not only humans, but here's an example of some viruses that are infecting a bacteria cell. And you can see these have a rather strange looking appearance. They're called bacteriophage, and they sometimes look like these lunar lander type things. So, okay. All right, so questions so far. Okay, whoops, wrong one. Okay, so I'm just going to start the next topic here just a little bit. I got about 5 minutes to go. I've got my time right. Yeah, okay. All right, so I'm skipping around a little bit here. Mostly I'm going in the order of the chapters in the book, but I wanted to jump ahead into this chapter on microbial growth because this matches up really nicely with what you'll be doing in lab on Wednesday and how you'll be kind of getting oriented to the ways that we grow up bacteria. So we're going to talk a little bit about culturing microorganisms, and just there's a lot of like terminology to get familiar with around that. So you're going to hear it from me, you're going to hear it from your lab professor if you're in lab, and it's okay to hear it a couple different places. Okay, so we're going to talk, you'll talk a lot about in the lab keeping cultures pure. So a culture is what we're using to grow the organism. It might be a test tube, it might be a petri dish. And what's pure, that just means that there's only one organism growing in there. So why do we want to do that? I guess we'll talk about that a little bit. But we're going to talk a little bit about some different kinds of media as well, and some terminology that we use there. Okay, so just beginning culturing microorganisms. Ever since we have learned to do this in the lab, the very first medias that we used were liquid. Okay, and we're going to refer to those as broths. That just means liquid media. Okay, and today, where does liquid media come from? Well, we buy it like this in powdered form. On that bottle, it says nutrient broth, and you weigh out the appropriate amount, and then you add it to some distilled water, and you maybe stir it up with a stir bar, but you're always going to heat it up to dissolve it to boiling, and heat it up long enough so that it's sterile. Okay. So what are broths? Basically, they're a collection of nutrients, and I'll give you some examples of that pretty soon. Nutrients, vitamins, minerals. Okay, to support growth of microorganisms. Okay, so liquid medias are going to be good to use if you want to grow up lots of organisms. They generally grow pretty quickly because they have lots of room to move around, and they're not inhibited by any kind of solid media. How do you tell if there are microorganisms growing in your broth culture? Well, it should look cloudy. So it's kind of hard to see there, but this first tube is the sterile media. By sterile, I mean no microbes in it at all. And you can't be sure by looking at it, but that looks pretty clear, pretty transparent. And then here's a culture that's starting to look a little bit cloudy. This one looks really cloudy. Okay, all that cloudiness, those are cells. And oftentimes that cloudiness will kind of settle out to the bottom. And you may need to kind of, we do a little kind of finger vortex. So you take your finger and you kind of hit that tube and it will swirl up from the bottom and you can say, oh yeah, there's my cells. Okay, so, you know, sometimes you're sure there's something growing in there, but sometimes it's not obvious. Okay, the disadvantage of using this kind of media is that you can't tell if your culture is pure. And remember, I didn't write that down, but if it's pure, there's one organism growing, and if it's not pure, mixed, there's lots of different organisms growing. So you can't really tell by looking at that because everything kind of looks the same. It's the same cloudiness, okay? All right, so we'll start next time. We'll talk more about solid media and different kinds of media, and then move on to microscopy. So, have a good day.
Professor, have a good day.
Yeah, you too.